Hi, this is session 3.1 of Biochemistry for Herbalists, and today we're going to have a look at photosynthesis. We'll see how photosynthesis is a major source of carbon for all organisms. Look at the dark reactions of photosynthesis, find out a bit more about chloroplasts, and then have a look at the light reactions. All will become clear. Most biological molecules are constructed from carbon to carbon covalent bonds. Where does that carbon come from? Mostly from the metabolic addition of carbon, carboxylation using carbon dioxide, CO2, and methylation using methyl groups, CH3. And when carbon is removed, we have decarboxylation and demethylation. Most of the carbon in terrestrial organisms has been fixed initially by plants. The chloroplasts in plants are able to fix carbon dioxide from the air using light energy to make carbohydrates. In the process, they split water and liberate oxygen, O2. The overall reaction of photosynthesis is carbon dioxide, CO2, plus water, H2O. Intervention of light energy gives glucose, C6H12O6, and oxygen, O2. So how come mixing carbon dioxide and water gives you sugars? Because if you mix carbon dioxide and water, all you get is fizzy water, really. But plants have a laboratory in a leaf called a chloroplast, which is able to take the carbon dioxide and fix it to make sugars. That process is called photosynthesis, synthesis or making using light. It's divided physically by the compartments in the chloroplast into light independent reactions, so-called dark reactions, and light dependent reactions, the light reactions. The dark reactions take place in the clear, colourless stroma or matrix of the chloroplast. They fix carbon dioxide onto other molecules. This is an energy consuming process. The light reactions take place in the pigmented grana or thylakoid membranes of the chloroplast. These trap energy from light particles, otherwise known as photons, and the light reactions are energy producing. The light reactions and the dark reactions are interdependent. ATP and NADPH are generated by the splitting of water in a light reaction. This production of ATP and NADPH powers the capture of carbon dioxide to form carbohydrates. So when we have a formula with N afterwards, there could be any number of multiples of that. So all carbohydrates contain this ratio of carbon, hydrogen and oxygen. First we'll have a look at the dark reactions of photosynthesis. This is a cycle of reactions that takes place in the stroma, in the juicy part inside the chloroplast. We'll have a look at a chloroplast in a minute just to see where we are, but in the meantime let's have a look at this reaction. It's in three phases. The first phase is the carbon fixation, and the starting point is this 5-carbon sugar, ribulose 5-phosphate. Has another phosphate added from ATP to make ribulose 1,5-bisphosphate. Then a carbon dioxide is added by an enzyme called ribulose bisphosphate carboxylase, otherwise known as Rubisco. This results in a 6-carbon molecule, which has a brief life before it splits into 2 3-carbon molecules, 3-phosphoglycerate. In phase 2, reduction happens. Reduction is opposite of oxidation, and in this case it means the addition of hydrogen. 3-phosphoglycerate is energized by phosphorylation by ATP, so it has a second phosphate added. This enables it to be able to oxidize the NADPH and itself become reduced, so producing glyceraldehyde phosphate, sometimes called GAP or G3P, and the phosphate is removed in the process. In phase 3 of the dark reactions, the Calvin cycle, the glyceraldehyde 3-phosphate is regenerated to ribulose 5-phosphate by a complex set of reactions involving 3-carbon, 4-carbon, 5, 6 and 7-carbon compounds. And if you really can be bothered, you can trace all these pathways through different size of sugars, producing in the end ribulose 5-phosphate. But not all the glyceraldehyde 3-phosphate is regenerated into ribulose 5-phosphate. Some of it goes to a reversal of glycolysis, gluconeogenesis, that remakes glucose, and then the glucose can be used by the plant for energy in a similar way to how it's used by other organisms, as we'll see in a later lecture about respiration. So some of the G3P goes to make glucose for respiration. So summarizing that dark reaction, it's a cycle of reactions called the Calvin cycle after the biochemist who discovered it, and the cycle is primed by a 5-carbon sugar called ribulose 5-phosphate. Energy from ATP and NADPH is used in the dark reaction to fix carbon dioxide from air and convert it into sugars. These molecules are generated in the light reaction. These dark reactions take place in the stroma of chloroplasts, and here's a cutaway of a chloroplast. You remember in the last lecture we were talking about organelles, and this is the organelle that's only found in plants, green colour because of the chlorophyll in the granae. This is an artist's impression, not a photograph or anything. So this is the stroma, the matrix, and you have lamellae, which are membrane complexes, and a stack of disc-like membrane structures called thylakoids. So the chloroplast is double membraned. The inner membrane envelops the aqueous stroma, and these I've already described these membrane complexes, and these are the site of the light reactions, in particular in this thylakoids. So if you take a cross section through one of those stack of coins, you can build a schematic diagram where you can see that embedded in these membranes, these thylakoid membranes, there's lots of protein complexes. Photosystem 1 and photosystem 2, the green ones and the blue ones, contain photosynthetic pigments, chlorophyll and carotenoids. There are electron transport molecules, cytochrome and plastoquinone, so these orange ones represent the cytochromes, and these carry out reduction-oxidation reactions. And finally, these club-shaped purple lumps represent ATPase or ATP synthase complex. So these are the players in the light reaction in the thylakoid membrane. So here's how it goes. We've explored it further. Here's the thylakoid membrane, the phospholipid bilayer, and here are the protein complexes embedded in it. Now, through reasons of history, photosystem 2 comes first in the sequence. Photosystem 1 was first discovered, but later they found photosystem 2, and in terms of the sequence of the reactions, photosystem 2 is the first where we begin. Light excites the chlorophyll and carotenoid molecules of PS2. This enables the oxygen evolving complex to split water into oxygen molecules and hydrogen ions or protons, because hydrogen atom has one proton, one electron. If you take off an electron, you're left with a single proton. Sometimes this oxygen evolving complex is called a photochemical reaction center. These protons are then released into the thylakoid lumen. So remember that this is the disc here, this is the inside of the disc, the inside of the thylakoid. This reaction, the splitting of water, is called the Hill reaction. Again, named after a biochemist called Hill. Meanwhile, electrons that have been split off from the water are transferred via plastoquinone to cytochrome B. So the blue dotted line is the path of electrons. Protons are translocated from the stroma because this is a reduction oxidation reaction. So protons are sucked in from the stroma and carried along with the electrons, and then plastoquinone gives up the electron to cytochrome and gives up the proton to the thylakoid lumen. And then the electrons continue the transfer to photosystem 1. Light energy absorbed by photosystem 1 boosts the energy of the electrons, and they're passed to a second electron transport chain, a second chain of redox reactions, which converts NADP to NADPH. So we're starting to get a concentration of protons in the thylakoid lumen from the splitting of water and from the translocation of protons from the stroma. So then we have what we call a proton gradient, or a pH gradient, across this membrane. So there's far more hydrogen ions inside the thylakoid than outside in the stroma. ATP synthase complex opens a special pore in the membrane which allows the hydrogen ions or protons to flow out of the thylakoid. This flow provides what we call a proton motive force, which provides energy for the creation of ATP from ADP and inorganic phosphate. ATP then goes on to be the energy currency molecule because it has a high energy potential. So in this way, the light reactions regenerate ATP and NADPH for use in the stroma by the dark reactions of the Calvin cycle in the production of sugar molecules from carbon dioxide and water. Photosynthesis in plants captures carbon in the form of carbon dioxide. It happens in chloroplasts. These are special organelles that contain light-absorbing pigments, membrane-bound enzyme complexes, and an aqueous enzyme-rich fluid called stroma. Dark reactions take place in the stroma involving an enzyme called rubisco, which fixes carbon dioxide to ribulose 5-phosphate. This the resulting product splits into 3 carbon molecules using ATP and NADPH. This can then be used to regenerate